Why the latest flu season was particularly nasty, why influenza keeps evading our seasonal vaccines. Are we going to ever come up with a single vaccine that protects us against all influenza strains? All these questions and more are going to be answered by my guest today, Dr. Florian Krammer, who is an associate professor at the Icahn School of Medicine at Mount Sinai, and he is an expert on flu vaccines. Welcome, Florian, to ASM Studio. Thanks for joining us.
Thank you very much.
So let's start from the beginning. Why the latest flu season was particularly bad? Any insights?
Well, there are a couple of insights, but before I start to talk about the problems that we have with the current flu vaccines, I want to actually state that the flu vaccines work and people should take them every year, everybody, okay? But obviously there's problems. First of all, the current vaccines that we have are not terribly immunogenic, and they only work when they are well matched to the circulating strains. Now that's one of the problems because influenza viruses change all the time, and they might even change when you culture them and when you make vaccines. And we assume that that is what happened in the last season, that the vaccines that we had were not very well matched to what was circulating, and on top of that we had a very bad H3N2 season. And H3N2 is currently really an issue because it causes much more morbidity and mortality than H1N1 or influenza B. So when we have this combination of a not well matched vaccine with an H3N2 season, that's kind of a perfect storm.
Can we explain to our audience how the seasonal vaccine is selected, the strains?
So the vaccine strain selection is complicated. Basically, the WHO has centers around the globe, and they monitor circulating strains. So they take samples from people who are sick with the flu. And then they sequence these strains, and they look at the antigenicity of these strains and compare them to the antigenicity of the vaccines and sera that were raised against the vaccines. Now, if they detect that the reactivity of the sera that they raised against the vaccines is not very good against the circulating strains, they know that there is a mismatch in the current season, or that there might be variants that might cause a mismatch in the next season, and so they're going to update the strain selection. Now the problem is that it's not always very clear what's going to dominate the next season. You might see very few mismatched viruses in one season, and you might say that we should keep going with the strain that we have, but then in the next season the dynamics might be different and the mismatched strain might take over. So it's basically a combination of surveillance and a well-informed prediction. But that's the issue, because we are trying to predict the future with this and we don't always get it right.
So that's the best we can do. So we have a challenge in selecting the strains, and do we have any challenges with producing the vaccine itself?
Well, flu vaccines are really unique, right? All the other viral or bacterial vaccines that we are using, they have to be produced once, then they are used, and they work. Measles or mumps vaccines are used for many, many years now. They never have to change. You just need to produce more batches of the same vaccine, right? Influenza is the only one where we have to reformulate every year, and obviously that's not easy. The WHO makes the strain selection in February for the Northern Hemisphere, and then basically it's a race to get the vaccine manufactured, because the vaccine has to be in hospitals, in doctors' offices in September, October, right? And so you have to do this in a very fast manner. And the problem is also that, yes, we select vaccine strains, but these vaccine strains might give less or more yield in the vaccine production process. And in the end, if you have strains that don't grow very well, you might even have an issue with supply, right? And with making the amounts that we need.
So these vaccines are produced in eggs, right?
So the majority of the vaccines that we are using right now are produced in eggs, which is a process that has been developed in the 1940s. But there is a minority of vaccines that are actually produced in cell culture, in MDCK cells, in dog cells. And then we also have now a vaccine that's based on recombinant hemagglutinin. And there are differences between those vaccines.
So do they mutate during the production, during the replication in eggs?
Yeah, so that was one of the assumptions why the vaccine effectiveness in the last season wasn't very good. There were mutations in the hemagglutinin of the H3 strain, which were probably caused by adaptation to the substrate, to the X. And that might happen with some viruses because the viruses bind to a receptor on human cells if it replicates in humans. And the avian receptor, which is obviously present in hens' eggs, is a little bit different. And the virus might change its surface glycoprotein so that it binds better to these receptors that are present in hens' eggs. But these changes might also cause antigenic changes that in the end then lead to a mismatch.
So, can we explain to the audience how the virus enters the cells and why we do care a lot about HA?
Well, yeah, I mean, the virus has two surface glycoproteins, two spikes on the surface. And one of them is the HA or hemagglutinin. And that hemagglutinin, it's a trimer. It looks like a mushroom in a way. And the cap of the mushroom has binding sites for sialic acid. That's the receptor on the cells. And so the HA binds to the sialic acid on the host cell, and then the virus is basically taken up into the cell by endocytosis, and that triggers the virus replication process. But if you have an immune response or antibodies that block that step, that bind to this mushroom, to the cap, and the cap can't bind to the host cell receptors anymore, you obviously neutralize the virus and you're protected.
So, if we can stop the interaction between the HA head and it looks like, can we imagine it like this microphone, for example? It has a head and a stem, right? And this head is the one that clicks into the receptor. And if we neutralize the virus, we're going to stop its entry. And that's how vaccines work.
That is how the current vaccines work. It's more complicated than that.
Okay.
Because there's more ways to actually stop the virus, right?
Okay.
So there's other types of antibodies that bind to other proteins or other parts of the HA, not to the head, but to the stalk. And this is a different type of immunity, but in a way you can also inhibit virus replication by targeting different proteins or different parts than only that cap of the HA.
So you told us that we have to update the vaccine every year. That's because the virus mutates and the HA structure changes. There is a new trend that we hear about in the media called universal flu vaccine, and that's in fact your area of expertise. Can you explain to us what is a universal vaccine? How does it work?
Yes, of course. There's not one universal vaccine. There are several approaches, and it's actually not that new. The idea is relatively old.
When did it start?
So if you're honest, it goes back to Thomas Francis Jr. and antigenic sin, and he actually proposed to use antigenic sin to kind of induce a broader coverage and broader immunity in humans. But there's, for example, a paper from 1982 where people already tried to make a universal influenza virus vaccine.
And that developed into something else that we know about today?
It did not, because at that point we didn't have the molecular knowledge, the structural knowledge that we have right now. So as you said, there's now a trend to work on universal influenza virus vaccines, and it's widely recognized that we would need something like that. So a universal influenza virus vaccine would basically be a vaccine that you give once or twice, and then you're protected from any kind of drifted seasonal strains, from new pandemic strains, even from zoonotic strains that you could acquire from, handling chickens on a live bird market like H7 and 9. Mm-hmm. That's the idea for universal influenza virus vaccine. And the concept was really, obviously there's different targets, but the concept of a HA-based universal influenza virus vaccine was really boosted around 2008, 2009, because the technologies to get monoclonal antibodies out of humans was getting so good that we got more and more of these rare antibodies, and people are now using those antibodies to basically do antibody-guided vaccine design. And what we learned with some of these antibodies, specifically after the 2009 pandemic, is that our body can make antibodies that broadly neutralize influenza viruses, not only within one subtype, but even across subtypes. Each and every one of us has some memory B cells that express an antibody that neutralizes H1 and H5, and H7 and all these exotic bird flus. The problem is only that the levels of these antibodies in humans are very, very low, and the immune system doesn't like to make them. That's the biggest issue.
So let's just go back one step. So we have so many types of this HA, right? 18 types, 18 subtypes so far.
It kind of depends how you count. 16 real ones and
Two from bats.
HA-like ones. Yes.
Okay. And the vaccines, the seasonal vaccines, include two strains, one that protects against H1 and another that protects against H3, and we have one for influenza B, right?
Well, we have now two for influenza B,
Two
so
for
for
influenza.
both lineages.
Quadrivalent.
Correct.
So how does the universal vaccine differ from a classical seasonal vaccine? Is it the same composition? Can you explain more about the conserved target and how neutralizing antibodies can protect against several types of influenza?
So I think we have to go one step back even
Please.
further. There are several targets for universal influenza virus vaccines. We have T-cell targets, we have internal proteins of the virus that really don't change much. They are relatively conserved, but they can be targeted by antibodies. They have strong T-cell epitopes. And there's vaccines now in development that are really focusing on inducing strong T-cell responses against the internal proteins. And then we have vaccine candidates that actually focus on a very small ectodomain of an ion channel that sits on the virus, M2E. And those vaccines also work through different mechanisms, but this M2 ion channel is relatively conserved, so they would also work against a broad range of viruses. And then obviously we have vaccines that are vaccine candidates that are based on this conserved stalk domain of the hemagglutinin. And there it kind of depends because not all of these antibodies cover all 18 subtypes. Some cover, so the subtypes are divided in actually group 1 and group 2 hemagglutinins. Some cover group 1 hemagglutinins, others cover group 2 hemagglutinins, and influenza B is a different animal too. But if we just talk about stock vaccines, we will probably need one trivalent vaccine with a group 1, group 2, and a B component. But you wouldn't change that vaccine all the time, and it wouldn't matter if, the strain you want to protect against is an H1 or a drifted H1 or an H3 or an H5.
So how do you stimulate the immune response to produce antibodies against the stem region?
Yeah, that's really complicated, and that has been the issue for many, many years now. So the ideal way of doing it would be to just cut off the head. So if you present the hemagglutinin with a head and the stalk to the immune system, it just loves the head. It will make antibodies and even more antibodies against the head domain, but it will neglect the stalk domain. It just doesn't like it. And so if you cut off the head, you get these antibodies against the stalk domain, but that 1982 paper that I mentioned did exactly that. And what happens then is that the whole structure falls apart. And these antibodies really bind to conformational epitopes, right, which are easily destroyed. But there are now approaches, at least for group 1 hemagglutinins, to stabilize these head-only constructs. So that's one way to do this. Janssen has a vaccine candidate that is based more or less on a soluble stalk domain. The Vaccine Research Center at NIAID, they have a nanoparticle that presents this stalk domain. And those are valid approaches that I think are going into clinical trials now. We also have a vaccine candidate. It's a little bit different. It's a little bit more tricky, but we basically force the immune system to refocus on the stalk domain. So typically, if you are exposed to a very new strain of or new hemagglutinin, like an H5 hemagglutinin, that your body has never seen before, you get a primary or de novo immune response against the head domain because it's so different from everything else you have seen. But the stalk of H5 and H1 is relatively similar. And what happens if you get an H5 vaccine or if you get exposed to one of these avian viruses is that you get a recall response, and memory B cells that bind to these cross-reactive epitopes are getting boosted. And we are using the same concept just with a little bit different constructs. They're called chimeric hemagglutinins. And if you give them sequentially, so they always have the same head stalk domain, but drastically different head domains, you basically refocus the immune response to the stalk domain, and you get very high levels of these antibodies.
And they protect lab animals against influenza?
Well, yes, mice lie. Ferrets might completely over-exaggerate immunity, but in animal models, so far it works very well. So we can basically protect against any kind of influenza virus, including avian influenza viruses.
Excellent. So where are we in this process? Are we going to see universal vaccines in our lifetime?
I'm a big believer in that, yes. So the problem is that developing new vaccines is really tough and it takes a long time. I mean, if you look historically, sometimes 30 years, 40 years, that's the time that you need to develop vaccines. It's a little bit faster now, but a lot of these vaccines enter clinical trials now. There are some that have been already tested in phase 1 trials. Some of these vaccines that focus on the internal proteins have been tested in phase 1 trials. Our vaccine candidate is now in phase 1, actually in a phase 1/2 trial. And as I said, VRC and Janssen are probably also moving into phase 1 trials. But it will take time to go into phase 2, go into phase 3. And in addition to that, we are not sure yet, and I think the regulatory agencies like the FDA are also not sure yet how to deal with that, how to kind of license these vaccines and because you can only claim what you can show in a clinical trial, right? If you have a universal influenza virus vaccine candidate, you probably have to test it in a phase 3 trial for multiple seasons, including seasons where there's a mismatch with the traditional vaccine. But then that's the only thing that you can claim. You can't call it a universal influenza virus vaccine, right? Because you have only tested it against seasonal viruses. So there's a lot of uncertainty about how these vaccines are going to get licensed, about the whole development process. So there's a lot of open questions, but I think for all parties involved, it's kind of a learning by doing.
Right. So one of the most problems with the flu vaccines, the seasonal vaccine, is that people don't go and take their shots every year. If we ever get a seasonal vaccine, will it provide lifelong immunity? Is it going to be just one shot? Do we have to take it every year?
Well, I mean that's one of the issues that we face with the current vaccines as well right now, right? We have to reformulate and re-administer these vaccines every year because the strains change. But the dirty secret is also that immunity wanes over time, right? So we're always told to get the flu shot relatively early because I work in a hospital. Everybody in the hospital needs to get the flu shot and they want to make sure that by the deadline everybody is vaccinated. But your immunity that was induced by these influenza virus vaccines is going to wane within 6 months. It's not going away completely, but the titers are going down towards the end of the season. So with these universal influenza virus vaccine candidates, we now have to make sure that we induce really long-lasting immunity. Because a universal flu vaccine that you give and then you only have immunity for 6 months or a year is of very limited use. I mean, you can still use it against pandemics, but it wouldn't help much against seasonal influenza.
Is it really realistic that you can produce the vaccine and distribute it in a case of a pandemic?
You mean the matched vaccines that we have
Yeah.
right now?
No, if we ever get a universal vaccine.
So in a way, that's a tough question. Right now, the government is stockpiling vaccines against viruses that have pandemic potential. This includes H5 strains, H7 strains, things that we know might be dangerous. And if you have the vaccine already, you can roll it out pretty quickly. Obviously not to the whole population. We don't have that number of doses, right? But you can vaccinate healthcare workers, you can vaccinate policemen and so on and so forth, right? So, but for a universal vaccine, if it gives you long-lasting immunity, it's a little bit different because you would start vaccinating people and over years you could build up high vaccine coverage and then you wouldn't need to rush and vaccinate people if there is a pandemic occurring.
So we need to have enough number of people vaccinated in a community. This is what we call herd immunity, right? Can you explain what herd immunity is?
Um, yes, I can explain that, but it's less relevant for
For flu?
influenza right now.
Mhm.
So um, basically, what happens with viral vaccines like or with viruses like measles or mumps is that if you vaccinate a certain percentage of the population, usually above 90%, the viruses have no chance of spreading anymore. So not everybody has to be vaccinated, but if the majority is vaccinated, we get immunity and the virus can't spread anymore. That was, I think, formerly known as herd immunity. Now it's called community immunity.
Community.
With influenza, that's tough because the viruses change so much. But again, with the universal influenza virus vaccine, we could uh, be in a situation where we can build up herd immunity high enough to, I wouldn't say eradicate influenza, but limit the spread of influenza in a community. The
Can we
problem,
ever eradicate it, by the way?
I don't think so.
Why?
Well, influenza B doesn't have an animal reservoir, so that might be hypothetically possible. I don't know if it's realistic, but influenza A has a huge animal reservoir. In fact, influenza A is an avian virus. It's not a per se a mammalian virus, and so I guess you would have to kill all the birds in order to eradicate influenza, and
Many other animals.
probably.
Yeah, yeah. Okay. I think we have time for questions from the audience. Can I take questions?
Why does like the measles vaccines and the mumps vaccine? Why do they only need one, like what once, as opposed to the flu vaccine where you need many?
So that's a great question. The honest answer is we don't know. But there's a more informed question. So people have done mutagenesis studies with the glycoprotein of measles, for example, and the glycoprotein of influenza. Measles also has a hemagglutinin. And if you do these mutagenesis libraries, influenza HA is very, very, very tolerant of mutations. You can even like have 5 amino acid insertions in the head domain. It doesn't really care about it much. You can't do that with measles. You can't recover live measles if you do that. So what we see is that measles viruses over time change very little. Although there is still some circulation, you don't see strong evolution, although the error rates of the polymerases are very similar, right? And you don't see that antigenic drift. And so it seems that it's intrinsic to the glycoproteins and how much change they can tolerate. And measles seems to be not very, very tolerant, and that might be one of the reasons. Another reason might also be that the measles vaccine is very effective, right? It's a live virus, it replicates in your body, it gives you very, very solid immunity in terms of antibodies and also in terms of T cells. Most influenza virus vaccines don't do that. So it might have many reasons, but those might be two of them.
Any other questions? We have questions in the back.
Oh, I think you get a present.
The universal vaccine, what it does take into consideration? It take into consideration the variation in the pathogen or the influenza, or it take into consideration that the immunity ran out. There is no many antibodies. In other words, I want to know if, when you give the universal vaccine, you don't have to always continue giving seasonally the vaccine, or you are just, it's like boosting the immunity. Oh.
All right, let me address the second question first. I mean, it's not clear how it will turn out, right? People might still use seasonal vaccines, and they might use universal vaccines in addition to that. I don't think anybody thought through that scenario yet. I think the idea is if these vaccines turn out to be very efficacious, at some point you might not need matched seasonal vaccines anymore. In terms of the first question, I think you were talking about the sequence variation in these conserved parts. So if you look at sequence variation there, the sequence variation is low. It seems that there's not much change over time, so they seem to be fairly conserved. But obviously, the question is, if you build up a very strong immune response in the population against these targets, are they going to start to drift, right? And that's something that we don't know yet. there is some evidence that, for example, with the stalk domain, if you make escape mutants in the laboratory against these antibodies, which we can do, we get viruses, but these viruses are not very fit. They don't like to make these changes there. And the stalk domain actually has a very important function for the virus. It has the fusion machinery, so that part completely rearranges. And you can imagine it's like a machine. If you change something drastically in that machine, it doesn't work that well anymore. But the answer is, over time, we don't know. We might induce drift there as well.
One more question.
Hello. My question has to do with the stock domain of the HA. So we know that when antibodies bind to the head domain, they prevent viral entry because now the head domain can't bind sialic acid. But if we have the antibodies binding the stock, how is that conferring protection to the patient?
Yeah, very good question. So there's two mechanisms for the virus in order that it needs to go through to enter cells. The first is it needs to bind to the receptor, and then it gets internalized into an endosome. But it's still in the endosome, and the genome of the virus is still in the virus, right? What happens then is that the endosome gets acidified, and that triggers that fusion machinery, a huge rearrangement of the hemagglutinin. And that is necessary in order for the virus membrane to fuse with the endosomal membrane. And only then can the genome of the virus be released into the cell and starts the replication. Now, the stalk antibodies bind to that fusion machinery and basically lock it into place. So the pH gets lowered in the endosome, but nothing happens. So it's basically a second step. But we know now that there's additional mechanisms that these antibodies use. They have a lot of effector functions, and you probably also get a lot of killing of infected cells that is mediated by these antibodies.
I think we have time for one more question.
We don't have food anymore.
Yeah. Sorry.
Hello. In terms of computational biology, do you know if there are any industry groups or universities that are particularly working on improving the algorithm for predicting which strains will arise next and how viral they'll be?
Yes. That's kind of a guessing game since, like, I don't know, 60, 70 years. So there's a lot of work ongoing right now. There are some groups, there's one group in Cambridge, Yoshi Kawoka's group in Wisconsin. I think the CDC is also working on that. So people are trying to predict what happens. You cannot do that just by modeling. You need some data too, but you can try to make escape mutants in the lab with polyclonal sera. You can try to do these predictions. And this might work short term. It's not going to give you one answer. You probably get multiple answers, which in practical terms might not be that helpful because you would have to include many more strains into the vaccine. The problem is long term, right? You can look at the trajectory of a virus and you might get the prediction right of where it goes next year, but it would be really hard to find out where it is in 5 years. And what's very hard is to make any kind of predictions about what the next pandemic is going to be, right? In 2009, in let's say February, if you would have done a survey among influenza experts and you would have asked them what's going to be the next pandemic, 99.9% would have said H5N1, and it wasn't H1N1. So it's really tough to make predictions.
So I think we ran out of time. Thanks, Florian, and he will be around if anybody has any other questions. Thanks for joining us, and enjoy the rest of the meeting. Thank you.